Welcome to Manifested Publishers.
Hello learners, my name is Stephen Kariungi. Today we continue with our discussion on osmosis, a subtopic within the topic cell physiology. During the last lesson, we had an experiment to demonstrate how osmosis takes place using a whisking tubing. So today I want us to have a different illustration of the process of osmosis. So we'll have a diagram, and in this diagram we'll have a semi-permeable membrane. As you can see, the semi-permeable membrane has some pores through which materials can pass through. And on one side, we shall have distilled water, and the other side we shall have a sugar solution. So the distilled water has only one type of molecules, that is the water molecules. So that's a region where there is a high concentration of water molecules. And then, of course, on the other side, we have low concentration of water molecules. So they are scattered. But in addition to that, we also have sugar molecules. The sugar molecules are larger. So we have, those are sugar molecules. And then these are water molecules, the small ones. On this side, we only have water molecules. And then they are separated. That is a semi-permeable membrane. And then this semi-permeable membrane has some pores. Is a pore in the semi-permeable membrane. So in this case, we are saying that osmosis will take place from where the water molecules are in high concentration to where the water molecules are in low concentration. So the direction of osmosis is like that. So the arrow shows the direction of osmosis. So that's the key. shows the direction of osmosis. So basically, we could use this to illustrate that osmosis takes place from where there is high concentration of water molecules to where there is a low concentration of water molecules. We can also say that osmosis occurs from the dilute solution to the concentrated solution through or across the semi-permeable membrane. And the semi-permeable membrane has some pores through which only the water molecules can pass, not the sugar molecules. Not the sugar molecules because of their size. The sugar molecules are of large size compared to the water molecules. Now, so from that, we can still come up with another definition of osmosis from the diagram above. We can also define osmosis as the movement of water molecules. It has to always be water molecules from a hypotonic solution to a hypertonic solution across a semi-permeable membrane. So in our case here, the distilled water is hypotonic. The solution that has a high concentration of water molecules, we refer to it as being hypotonic, while the sugar solution is hypertonic, the one that has a low concentration of water molecules. So we can also say that osmosis takes place from a hypotonic solution to a hypertonic solution across the semi-permeable membrane. So on this, we can say that a hypotonic solution has a higher concentration of water molecules than the surrounding solution. Whereas a hypertonic solution has a lower concentration of water molecules, than the surrounding solution. Now, if the two solutions have the same concentration of water, then there is no osmosis that will take place. There is no osmosis if they have the same concentration of water molecules. And that's why we say that osmosis takes place until the solutions have equal concentration, or the solutions are at equilibrium. So in that, we can say that when the two solutions have the same concentration of water molecules, they are said to be isotonic. So none has a higher concentration than the other. They have the same concentration, the two solutions that are surrounding each other. So they are said to be isotonic if they have the same concentration of water molecules. And therefore, no osmosis takes place at this point. So that is basically an explanation on hypotonic solution, hypertonic solution, and isotonic solution. So we are going to have an assignment on the same. So the first question, what is A, hypotonic solution, B, hypertonic solution, C, isotonic solution, and D, sorry, we don't have D, and number 2, define osmosis using hypotonic and hypertonic. So we'll stop there. Until next time, goodbye.